To help us understand the concepts of phenotypes, genotypes, and their ratios from different crosses, we can use a concept called a Punnett Square. A Punnett Square is a simple tool that will help us visualize the ratios from these crosses. Let's start by using a simple example. We're going to start by using the example of eye color. And let's say, for simplicity's sake, there are two alleles for eye color. One, a capital B, which is a dominant allele that codes for brown eyes. The lowercase b will code for blue eyes. So an individual heterozygous for this trait will have brown eyes because they will have one big B allele and one little b allele. Let's start by crossing an individual with brown eyes, the father, and an individual with blue eyes, or the mother. So to start our Punnett square, we start by making a tic-tac-toe board. Then we divide the first cell up top, and we're going to put our father in the top and our mother in the column. Then we split up our father's alleles and put them in the top row, a big B here and a big B here. We then split up our mother's alleles and put them in the first column, a little b and a little b. To finish our Punnett square, we simply drop down one allele from the father and bring over one allele from the mother, like so. With this, our four offspring are in the cells that we just completed here. In this example, all of the offspring had the genotype big B, little b, which means that all offspring will have brown eyes. In this case, the genotype ratio is fairly simple because we only have one option and one phenotype. Let's cross, as a second example, two heterozygous individuals with genotypes big B little b and big B little b. Because they're heterozygous, they will express their dominant allele, which means that both of these individuals have brown eyes. Once again, we'll start with our tic-tac-toe board. We'll divide our first cell. We'll put our father here and our mother here. Then we split up each parent's alleles. Big B, little b, big B, and little b. To complete the Punnett square, we once again bring over our mother's alleles and we bring down our father's alleles. Now this is a slightly more complicated example and we get different results. One individual has a genotype of big B, which means this individual will have brown eyes. Two individuals have the genotype big B, little b, which means they will also have brown eyes. And the last individual has a genotype of little b, which means this individual will have blue eyes. It's interesting to note that although both parents had brown eyes, they had a child with blue eyes because they were carriers for this blue-eyed trait. From this, we can also see a difference between the genotype and the phenotype ratios. Our genotype ratio is 1 to 2 to 1 because we have one individual homozygous for the big B allele, two individuals who are heterozygous, and one individual who is homozygous for the little b allele. Our phenotype ratios, however, are slightly different. We have three individuals with brown eyes and one individual with blue eyes. And so we have a difference between our genotype ratio and our phenotype ratio.